Hello, welcome to the US HUPO educational series on spatial omics. So today we have a really interesting, we're going to have a really interesting discussion on quantitative imaging. So we're going to hear first from Reed Grosclose, who's the bio director of imaging at GSK, and then followed by Stephanie Colonia, who has worked out some strategies for starting quantitative imaging and performing quantitative imaging in her lab. So Kristen, can you introduce Reed, please?
Sure. There we go. Get my video working. So it is my pleasure to introduce Reed Grosclose. He works in industry. He comes to us from GSK, where he is director and fellow of bioimaging. He earned his BS in chemistry from the University of Tennessee in 2004, and his PhD in chemistry from Vanderbilt University in 2009 in the lab of Richard Caprioli. In Richard's group, he focused on exploring the utility of imaging mass spectrometry and on-tissue proteomics for the analysis and classification of cancerous tissues. He joined GSK in 2009, where his research interests have centered on the application of imaging mass spectrometry to assess the tissue distribution of drugs and their metabolites. His group at GSK is particularly interested in using imaging mass spectrometry to quantify and localize tissue concentrations of drug candidates and investigating mechanisms of pharmacology and safety. And with that, go ahead, Reed, take it away.
All right. Thank you, Kristen, and thank you, Peggy, and the organizers for this opportunity to present to you on some of the work that we've done at GSK using primarily imaging mass spectrometry. So as Kristen mentioned, I lead the MALDI imaging mass spectrometry group at GSK. We're in the bioimaging department. And today I'd just like to share with you some of the work that we've once again done using imaging to help drive decision making in drug discovery and development. Hopefully you can see my screen. Just ping me if you can't. But first of all, a little bit of background to kind of set the stage for the talk. So I think it's typically well recognized in drug discovery. The process is quite challenging, where we may start with a huge number of potential molecules that are shown to have some potential impact on a specific target or mechanism. But over the course of many years, through series of optimizations of potency, preclinical testing, and eventually testing in humans, most of these initial starting compounds will be filtered out, and the cost of progressing compounds that eventually fail is quite substantial. So there's a big push to better understand early on, obviously, which drugs might have potential to advance into humans, and which of those may have issues with whether it's efficacy or safety or any other number of factors that could limit progression. So there's kind of jargon that we hear a lot within our industry around the pillars of drug candidates. So it's been termed the 3 pillars of survival, and these are pretty basic conceptually, and I think make sense in terms of what you would expect from a drug. But one of the main factors that is critical is evidence that the drug does actually reach the target tissue or target cell, or any number of layers that you could investigate this. But obviously, in order to enact an effect, the drug must reach the target. Does it bind the target? Obviously, there's a lot of kinetics and various parameters around binding, but binding to the target is critical. And then through binding to that target, does it have the desired modulation or pharmacological effect that would give you this response, whether it's inhibition or any other potential pathways or factors that could drive this. So the drug candidates that meet these criteria, as you might expect, would have much greater potential to progress and become an actual medicine in humans. So sorry, let me rearrange my screen here. There we go. So the importance of spatial context. Historically, in terms of measuring the concentration of drugs in tissues, the primary approach would have been homogenization. So the tissue of interest would have been homogenized and analyzed by any number of methods, typically some sort of LCMS approach. But I think what's lost there is the spatial context of that drug distribution. So in many tissues, for example, a tumor, obviously there's a very heterogeneous, complex distribution of different cell types. And understanding the spatial context of that distribution of a drug can be very important in understanding the whatever its efficacy here. Is the drug actually reaching the tumor cells? Is it not? Any number of factors, but the spatial context can be critically important. So another area would be safety and toxicology. So when a pathologist is reviewing tissues from a toxicity study involving a drug, and there's a finding or an abnormal observation in a tissue, then the spatial context of drug distribution or drug metabolite distribution can be critical in helping assess kind of the risk of this toxicity, and understanding, is there a relationship between the drug metabolite and this tissue damage? So we primarily use imaging mass spectrometry to measure drug distribution. And just briefly, for those of you not familiar, we would start typically with a fresh frozen tissue, generate thin tissue sections, mount these onto a microscope slide. Our group primarily uses MALDI, but there's other approaches that use a similar concept to create ions into the gas phase. With MALDI, a laser. There's first some sample preparation steps, which I'm skipping here, but once prepped, the laser is then fired at the tissue surface. Mass spectra are acquired in a discrete grid across the surface of the tissue, and then once completed, the distribution of any of the detected ions could be visualized as an image based on their respective intensity. So it's quite powerful in terms of its versatility, its sensitivity. It's using the mass spectrometer as the detector, so you can kind of harness that ability to have high specificity. Any number of range of different species can be detected depending on your sample preparation workflow. It's label free, and then, as I'll get to later, it's also quantitative. So we also use a kind of a suite of supporting technologies: LCMS, histology, optical imaging, and then the data analysis is which I won't really get into much today, but it's absolutely critical because the volume of data that you can generate in these studies is quite extensive. So additionally, multimodal imaging, so using various different technologies for imaging distributions or imaging the anatomy or whatever it might be, but typically we find that contextualizing the ion images generated by imaging mass spectrometry, the added value of some of these other approaches that you could also use to measure various aspects of a tissue can be quite powerful. So this is kind of an older study, but I think helps frame kind of the power of this approach. This is looking at a section of fibrotic rat liver. This was kind of an approach. I won't get to the details of the study, but the idea was to generate this hepatotoxicity in rats and look at the distribution of a drug, but also some of the endogenous changes that have occurred. So in this study, sorry, I'm moving this part. I can't see my screen. There we go. Using multi-imaging, we can first look at the exogenously, the dose drug compounds. So in this example here in orange, it's the dosed compound, the verapine, but also we can measure one of the primary drug metabolites in the verapine, which is a 12-hydroxy species that's localized to this large portal vein area. So In addition to the drug, the exogenous, we can also measure the endogenous. So we can look at various endogenous components, some of the bile acids, various adenosine monophosphate, looking at kind of a hepatocellular distribution here. We can basically recreate the histology from these endogenous molecular species and correlate that with the distribution of the drug and really generate some very powerful kind of molecular histology information. So getting into a few examples of how we've utilized this technology over the past several years. This was an example where this was a safety, this was a toxicity study in juvenile rats for a drug called dabrafenib. And during a toxicity study where an intentionally high dose is administered to the rats to kind of understand your safety margins. So there's a background that's the purpose of these studies is once again to understand the tolerable levels of drug that can be achieved in animals and then kind of translate that to humans. But so this was an intentionally high dose, but what the pathologist found in reviewing the tissues was that many of the animals were observed to have these intratubular crystalline deposits distributed throughout their kidneys. And the question to us was, can you help us understand the composition of these deposits? So it was important for them to know, were these deposits primarily comprised of the dose drug, dabrafenib, or something else? So we did a large set of images and investigated the distribution of both dabrafenib and the major metabolites of dabrafenib. And what we initially found was that we did not observe any direct association of the dosed drug compound or its major metabolites with these crystalline deposits. We did, however, observe these calcium phosphate species directly associated with the crystalline deposits. So we could present back to the team that number one, we did not observe any evidence of drug-related material in these deposits. However, we do feel they're primarily comprised of calcium phosphate. And this helps kind of hypothesize different mechanisms of this formation. And in this case, there was kind of a risk assessment that the safety team could put together, which I won't get into the detail of here, but it basically involved in the juvenile rats, there was the primary excretion of dabrafenib was the liver hepatobiliary that was underdeveloped. It was causing a huge load of the drug in the kidney tubules. It was damaging these tubules, disrupting the calcium homeostasis, and then resulting in this deposit formation. But this information as to the composition was critical to help them start to build this hypothesis of various mechanisms that could be driving this. So to contrast this, the left is the rat kidney, which I just kind of went through. We had a separate study, similar finding, crystalline deposits within the tubules of a kidney. This was in mini pig, once again, a whole different drug. But in this case, interestingly, when we analyzed these areas with the deposits with MALDI imaging, we observed that these deposits appeared to be almost entirely comprised of the dose compound and its metabolites. So two unique scenarios, and I think good examples of where really imaging is required to really help contextualize these observations and understand the compositions of these deposits. So very briefly, an additional study we did in this is in rats as well, looking at the potential for ocular toxicity. This is a compound retigabine that was on the market for epilepsy. Excuse me. And there were some findings in humans that range from abnormal skin pigmentation. So there were patients that were administered retigabine for many years, and they had kind of a bluish coloration of some of the mucosal tissues, fingernail beds, lips, and the FDA was very concerned about the risk of ocular toxicity, because there was a thought that some sort of melanin binding mechanism was driving this. So there was a huge series of different studies, investigations that were initiated as a result of this. We contributed to one looking at this ocular toxicity in rats. So just to kind of summarize, there's a lot more behind this, but just as a general summary, what we observed was that, well, first of all, what was known was that retigabine was forming a dimerized metabolite, and this was known to occur in vitro. It had been observed, and there were others, studies that had looked at the homogeneous concentration of this dimer metabolite in eyes, but really what they needed was the spatial context to better understand what was going on. So we did an imaging study. What I'm showing here, I don't know if you can see my mouse, but in the bottom left is the distribution of this dimer metabolite throughout a cross section of the rat eye. If we go to the middle part of the screen, at higher spatial resolution, we could kind of zoom in on a specific layer of the rat eye, the choroid, which is the pigmented melanin containing a highly vascularized layer of the eye, and we could see that this metabolite was very highly localized to this choroid layer. On the right hand side, we could zoom in even further at 5 microns spatial resolution, and once again observe this choroid distribution of the dimer metabolite, but also we could investigate further what looked like it did not appear that let me see if I can add a laser pointer here. Sorry. Maybe you can see that, but if not, it's okay. On the far right, this panel B, and I've got it labeled in panel A, the RPE, the retinal pigmented epithelium, is part of the retina. The choroid is not. So we did not see evidence of this dimer metabolite accumulating in the RPE. So this was important because this region, this is the region, the retina obviously is the region of eye containing the photoreceptor. So ocular toxicity would have been a much greater concern had we seen it extending into that region. We could kind of further establish this by looking at one of the retinoid species, which was very highly localized to the RPE, and we could show that we did not really see any overlap or penetration of this dimer metabolite into that retinal pigmented epithelium. So once again, just another example of the importance of the spatial context. I don't have the values here, but from LCMS of eye homogenates, the concentration that was observed of this dimer metabolite was relatively low. I believe it was in like the 50 to 100 nanograms per mil. But what's interesting is that with imaging, it's important to establish that that concentration that was observed from a homogenate obviously is not evenly distributed throughout the volume of the eye. It's very highly localized within a very specific region. So I think, it kind of adds important context to that value by showing that concentration, once again, is the local concentration is much higher. That average of the whole eye could be deceptive. So we've spent a significant amount of time in our group over the past several years trying to establish, quantitative workflows such that we can kind of go beyond just looking at the distribution, but also, report these ion images with quantitative information. So we started, well, the basic concept of what we term the tissue mimetic model is that we create aliquots of a homogenized tissue sample. Typically, we use rat liver. We homogenize this tissue with a bead homogenizer such that you can actually pipette aliquots and without adding any additional homogenization solvent. We would then spike these homogenates with a known concentration of the drug, analyte of interest. and then freeze these homogenates into a cylindrical mold. This then allows us to, once frozen, we can then collect thin sections of the cylindrical mold. We can mount those adjacent to our tissue of interest. And then, this slide undergoes the same sample preparation and imaging acquisition conditions, such that upon generating the data from each layer where we have a known concentration, we can then construct a calibration curve, and we can then extrapolate. We can basically quantify across the whole tissue level. We can go into specific regions of a tissue, and based on this calibration curve, we can start to define or assign quantitative information. So we've gone through several iterations of this, and we've recently kind of adapted this, what was kind of a tissue microarray approach. So Jeremy Berry just really did a huge amount of work in establishing a lot of these quantitative workflows, and he's no longer in the group, but before he left, he kind of built this workflow, which was based on a previous publication using a similar approach. But basically in this approach, the concept is the same, but it starts to get around. One of the limitations of this approach was we found it limiting in terms of the number of layers that you could effectively create in this mold, and that limited the number of calibration points you could establish. So with the tissue microarray approach, it really opens the door to include many more calibration points. This can be critical for a number of reasons in tissues where you have a very broad range of concentrations. It can really help better establish your limit of detection, and just has a number of advantages. So this is kind of the approach that in our group we've moved towards adopting. But so just to kind of give you an example of the utilization of this tissue mimetic model approach in a recent study. Once again, this was a huge study completed by Jeremy, which I'm just kind of touching on the highlights here, but there's a lot more behind this. So the idea, or sorry, the basis of the study was to look at the well, this was centered around a program that was developing medications for treating brain tumors, primary metastatic brain tumors. So there were two compounds of interest in this study that the team was interested in differentiating the distribution in brain. Here, I'll refer to them as GSK1 and GSK2. And I think it's pretty well established that this brain tumors, remain a critical unmet need for patients, and the prognosis is terrible, very low 5-year survival rates. But a lot of the issue is the inability to penetrate the blood-brain barrier. So the blood-brain barrier obviously has a protective effect and intentionally limits the penetration of many compounds. So various efflux transporters can make achieving therapeutic concentrations of various drugs in the brain very challenging. So this study was in mice, and it was using a metastatic, a brain metastatic mouse. It's a tumor model. So this was a human breast cancer cell line which had been transfected with luciferase. The mice received an intracardiac injection, and then over the course of around 30 to 40 days, using bioluminescence imaging in vivo, the progression of the implantation of the tumor cells in the mouse brain was monitored. And so the way the study proceeded was there was a large cohort of mice that initially received this intracardiac injection However, only a fraction of the mice actually developed brain tumors. So it's maybe 10, 15, 20% that actually go on to develop the brain tumors. Others just simply do not. So by tracking this brain tumor development with the bioluminescence, various groups were annotated to look at different factors. So there was a group of mice that did have brain tumors that were administered one compound, GSK1. This was a 4-day oral dosing. This group had 4 mice. Then there was a second group that, based on this bioluminescence review and establishing, specific mice which did have brain tumors, there was a second cohort of 3 mice given GSK2, a separate compound. And then from the mice that did not develop brain tumors, the groups of labeled here, no brain tumors, were created. And then finally, there was a 5th vehicle control, which did not receive cells. So from each of these mice, following the 4 days oral administration of each respective compounds, the brains were collected and plasma was also collected throughout the study for LCMS quantification. So just really quickly, the sectioning strategy that Jeremy took with this was for each of the brains analyzed, 5 distinct coronal planes were targeted, and from each of those planes, 3 serial sections were collected for imaging mass spectrometry. So 5 distinct planes, 15 sections per animal. A serial H&E was also collected from each IMS section for the histological evaluation. And then the trimmings, the tissue trimmings between all of the IMS sections were collected for each animal for kind of a bulk homogeneous LC mass. There was a histology group that confirmed in sections that contained the tumor just to that these cell types observed were of human origin. So mitochondrial marker. And once again here you can see kind of an example of the H&E, but I'll kind of get back to these in a second. So kind of getting into the results here, what we saw was quite a significant difference between the 2 compounds in terms of the level of brain penetration. So GSK1 generally showed much more extensive penetration into the brain. On the left here, this is looking at an animal that did have brain metastases, administered GSK1, brain metastases, administered GSK2. These were animals where no brain metastases were observed based on the bioluminescence. So once again, this column was administered GSK1, GSK2. So between these 2 groups, once again, it's pretty clear that the GSK1 compound was distributing much more extensively into the brain. Once again, we did the plasma measurements. We also did the bulk brain homogenates. And then from each of the sections analyzed by MALDI imaging, we created a brain section concentration. So a couple of things to point out here. Number one, the correlation between the MALDI imaging quantification and the bulk brain homogenate was there were minor differences, but generally speaking, were quite consistent. And once again, established that the levels in the brains for the animals administered GSK2 were quite low. And there were also pretty significant differences in the plasma levels achieved between the 2 compounds. And so by using this KPUU, this is the unbound brain to plasma drug partition coefficient, which kind of gives you insight on the level of brain penetration, and looks at the free drug, which I won't get into the details of that for the sake of time, but was able to show, just generally speaking, even when adjusting for the plasma, the GSK1 compound achieved much higher brain penetration. So we did not observe the second compound GSK2 in the brain, and even though we had what I would say was really good limit of detection for these 2 compounds, GSK2, based on the LCMS, the levels were just incredibly low, so we did not see it. But what you can see here, and I'll show you in the next slide a little bit more detail. So once again, this column is the mice containing the brain metastases. and you can see that the primary distribution was in both the ventricles. Maybe you're more clear here where there's no tumors, but then we also had these areas that were clearly tumors, which I'll kind of get into in this next slide here. But so digging in a little bit more detail in the animals, this is looking at kind of representative ion images for a single animal. Once again, there were 5 coronal planes, 3 replicates from each plane. Here you can see that from this second panel ion image, there's a very clear brain tumor observed, and then we can see using imaging this relatively high levels of this drug penetrating into this tumor region. And so, you know, we can even get to the point where we can assign absolute quantitative values to these very specific regions. The table on the right-hand side is the summary of all of the data points captured across the 4 animals, so 15 sections from 4 different animals in this group, so a total of 45 brain sections imaged, and just kind of plotting the distributions here between 3 histological regions: the parenchyma, the tumor, and then the ventricle space. And then on the right, just for kind of reference, is the whole tissue section concentrations. And I think what's important here is just showing the, you know, sometimes the whole tissue, which would be representative of the whole brain distribution, it can be not misleading, but I think it's important to add the context of these localized concentrations. So here you can see the tumor concentration is the mean of that is 5-fold higher than the whole tissue section average. So just to kind of move on for the sake of time, just to summarize here, this is where differentiating these 2 compounds, that spatial information becomes critical. We could show effectively that the penetration of GSK1 versus the second compound was much higher. High levels of tumor penetration from GSK1, and what's really critical here is that localized quantitative assessment. So that opens the door for kind of some advanced modeling of the tumor drug penetration kinetics. You can imagine things like it's understood maybe the efficacious concentration needed. And so now we can start to provide this very detailed quantitative spatial information back to teams and really help them have a deeper understanding of some of the mechanisms of action distribution. So I'm going to skip through. I want to keep us on time. I think I'm probably out of time. So I'll go through these really quickly, but just to show some additional applications that we've started utilizing and kind of some unique targeted drug delivery approaches. Small molecule drug conjugates. Using imaging, we can track different pieces of these drug conjugates. So there's kind of a linker, then there's a payload. And once again, with mass spectrometry, we can easily distinguish these pieces with different masses and start to assess kind of delivery and cleavage and a number of different factors. We've also done some work with antibodies that are tagged with both a fluorescent label and, in this case, what's called an antibody recruiting molecule, so a small molecule linker. Then in vivo studies, we can do this kind of multimodal approach where with MALDI imaging, we can image the antibody recruiting molecule, the small molecule piece of this conjugate, and then with fluorescence, we can measure the antibody distribution, so which can really provide some powerful information on how this distributes and the kinetics of that. So let me see, 30 minutes. Sorry. Okay. Yep. I'll tell you what. Just keeps on track. Let's skip these and. I will finish up there. I'll thank the team and thank everyone online for your time, and happy to answer questions once we get to that point. So,
thank you so much, Reed. That was an amazing talk. I have questions for later, but we're going to save those. So if you want to stop sharing, we'll move to our next speaker. So our next speaker is Dr. Stephanie Colonia. She is Associate Professor of Chemistry and Associate Vice Chancellor for Research at the University of Illinois. She received her BS in Chemistry from the University of Arizona, followed by doctoral studies at Texas A&M under the mentorship of David H. Russell. So she has developed a research program focused on integrating mass spectrometry-based proteomics and lipidomics to understand neurodegeneration, particularly due to the lysosomal storage disorder through the Niemann-Pick type C protein pathways. Stephanie?
Thanks so much, Peggy, for the opportunity to be here. And thank you so much to Reed for this great overview of all of the capabilities that mass spectrometry and imaging modalities can really inform studies. So what I wanted to do was take this in a little bit of a tutorial-like manner and think about the concept of, you know, how are you going to actually carry out this imaging study? The screen seems to be frozen. Let's see here. All right. So you've learned how to image at this point. Now what? And so what I'd like to propose is that there are some aspects of the experiment that you really want to think about as you're trying to drive your project into a quantitative imaging platform. So things that will be really important to be considered are reproducibility, whether or not you need to carry out your study in a relative quantification mode or an absolute quantification mode. And I think that Reid really highlighted some of the power of both of those strategies. And at this point, I'm under the assumption that you've characterized the signal and you know the identity. So we know where it is based on our initial imaging experiments. Now, how much is there? So to do that, you must consider a few challenges that are associated with the quantification study. Things such as the matrix effects, including ion suppression based on the chemical environment, how you might normalize the data set. There are a number of different strategies, including the total ion current, whether or not you have an internal standard or an authenticated standard, or the tissue extraction coefficient method, for example. And additionally, you might want to consider whether or not you need to have any specialized sample preparation, such as matrix additives or derivatization strategies. All right, so with those all in mind, I'd like to share a few examples of how we've used mass spectrometry imaging to study a rare neurodegenerative disease called Niemann-Pick type C. So Niemann-Pick type C, or NPC, is a fatal autosomal recessive neurodegenerative disease. It's biochemically characterized by the accumulation of cholesterol and glycosphingolipids in the endolysosomal system. It's genetic in nature and therefore arises due to mutations in either the NPC1 or the NPC2 genes. However, 95% of clinical diagnoses are due to NPC1 mutations. Shown at the bottom is a series of skin fibroblasts that have been stained with philippin, which will bind unesterified cholesterol. And hopefully you can appreciate in these three different skin cell cultures, this distinct puncta that are stained in green, and this represents the cholesterol storage in the lysosomal compartments relative to the control on the left. Now, you may also notice that above each image is a different genetic mutation, and in fact, there are over 300 clinically relevant genetic mutations for this disorder. Now, thinking about beyond the biochemical aspect and thinking about the clinical manifestation, there's actually a large variation with respect to age of onset and disease severity. And if you focus on just the brain, these include ataxia, progressive dementia, seizures, and supranuclear vertical gaze palsy, or the inability to rapidly move the eyes vertically. And there's currently no FDA-approved therapy for NPC. However, a subset of individuals do take a drug called miglustat, which is approved for Gaucher disease, and this molecule 2-hydroxypropyl beta cyclodextrin has been in clinical trials. So with regard to focusing on the neuronal phenotype of the disease, there's actually very clear neuron loss in both the cerebral cortex and the cerebellum. And really the hallmark of this disease is this cerebellar degeneration, which is shown in the image on the right. These are sections of the cerebellum. On the top is a wild type animal. The bottom is one that lacks the NPC1 gene. And the red is staining for a protein called calbendin D. And calbendin D is a marker of Purkinje neurons. And so hopefully you can appreciate that if you look at the cerebellum from the front to the back, there's distinct loss of these Purkinje neurons, particularly in lobules, let's say 1 through 8. This happens in a patterned manner. So if you were to take a snapshot earlier in time, you would actually see the preservation of the Purkinje neurons here in these later lobules. By which these neurons die is not well understood. There is some overlap with other common neurodegenerative diseases such as Alzheimer's disease, but this is a distinctly different disorder. And I should add that while we focus mostly on the brain in my laboratory, there is significant liver disease that's also affected. Now, you might then question, much like I did some years ago, if you have this cholesterol storage or sphingolipid storage, what do the distributions of lipids look like? And so this is a study that shows here on the right in whole brain lysates a number of sphingolipids that are altered in the mouse model of NPC disease. And so this is the wild type at 3 different time points here on the left, the heterozygous carriers on the right, and then the far right is the actual mutant animals. And you'll notice that, depending on both the time point and the sphingolipid species, there are distinct patterns of up and down levels in the whole brain tissue. So this kind of brings to light this question, but what do the distributions look like? And that's really where the power of being able to image using mass spectrometry comes in. So when I started my lab in 2015, I really wanted to perform this mass spectrometry imaging experiment. And so just recall that we have this progressive neurodegeneration here on the left. And the question is can we get lipid signals that may tell us something about why this happens or that change with the progressive neurodegeneration? And so some of the very first images that we collected are shown here on the right, which are have been analyzed by tick normalization. So again, this is the cerebellum. And these are just 2 examples. On the top panel is the diseased animal. The bottom panel is the wild type animal. And so for these 2 molecules, the lipid PIP2 and the lipid GM2 have very distinct patterns and distributions, as well as accumulations or loss. Now, going back to this concept of reproducibility, we wanted to ask the question, which is, what is the image-to-image variability in our hands? As you may have kind of picked up in the introduction, my background is primarily in protein-based mass spectrometry. And so imaging was rather new to us, and we really wanted to make sure that we were doing things in the most robust manner. And so to look at that, what we did was we developed this workflow to look at the reproducibility of our imaging acquisition. And I'll share this example with you, which addressed some of the questions that we had at the time, but I should also note that there are some caveats, and this is something that, must be kind of updated. There are limitations to every tool, and this itself also includes its own. So the idea that we did was that we took a region of interest, and we then took the most abundant signals, and in this case, we picked 40 for this example. We aligned the spectra from multiple imaging runs by a technique called QT clustering and converted to unit vector, and assessed the similarity using the dot product analysis. Ultimately, we are then able to extract mass to charge and normalized intensity to generate something called a consensus spectrum as a readout of all of the experiments in one, and then we're able to make our assignments. And so by doing this iteratively, we're both able to see how similar each run is to the collective group, as well as to see how reproducible or how different the genotypes of the control versus the disease look like. So in our very first study, what we were able to do is we were actually able to look at so 6 control animals, 5 mutant animals, and we did serial sections to look at various reproducibility, although you wouldn't want to go too far. In this case, we tried to stay right at the midline. And so we had something on the order of 13 to 17 data sets or images that we were able to look at. And so then if we use the 95% cutoff and looked at the correlation between each individual replicate to the consensus, we were able to establish essentially whether something passes or fails. And ultimately, we're able to generate then a consensus mass spectrum that's shown here with the disease animal at the top and the control animal in the blue on the bottom. Now, what's important to note is that this is the consensus spectrum for the entire region of interest. In this example was the entire cerebellum. But what we are able to show is that there are certainly intensity-based differences. And so going back to that very first image that I shared with you, what I can say is that the triangle represents this ganglioside GM2, which indeed is known to be elevated in the disease. It's shown here in our image on the right. And the square represents this lipid PIP2, which was a phospholipid that had not been previously reported to be altered in the disease and really opened up a new area of research for our research program. Moving forward, we can actually look at the entire data set. So those were just some select analyses, and we collected the data really in 3 kind of segments, and that was due to limitations of the data collection and file sizes that we had in our laboratory. But essentially, if we look at the higher mass ganglioside region in negative ion mode, we look at lower mass in negative ion or positive ion mode. We're able to, again, look at the dot product of the consensus spectrum of the control versus the disease. And so from these data, what we're able to say then is that for B and C, there's actually quite a bit of similarity in terms of the signals that are observed, whereas in the ganglioside or in panel A, the ones that I just showed you are slightly different. And so this gave us a tool to be able to start to look at differences among genotypes for our imaging analyses. Now, I told you that you can start to think about how to do quantification, and I think Reed alluded to this in his talk as well, which is that oftentimes people will do some type of secondary analysis, like a homogenate, and do LCMS, and in this case, that's exactly what we did. So following total ion or TIC normalization here in the imaging experiment on the left, where we show that there's this decrease of that lipid PIP2. We then used extracts to confirm that indeed we see that. But of course, what's lost is some of the granular detail of the spatial localization. So if you start thinking about doing a mass spectrometry imaging experiment, a recent study has kind of proposed some of these good practices for imaging. And so these include reporting things such as your step size and your spot size, the number of pixels that might be of your study, as well as something about the assignment and the confidence of your annotation. Of course, if you are working with a drug distribution study or that, you would have high confidence based on what you've introduced into the system. But if you're doing this in more of a discovery-based mode, then this would be certainly improvement in confidence whether you have tandem and mass data. Other recommendations include reporting the mass resolution or resolving power of the instrumentation that was used, as well as the time of the acquisition. So going forward, we can think about a couple of different models for actually using internal standards for quantitative mass spectrometry imaging. So this is a nice review that was published by Ralph Tobias a couple of years ago, in which he reported, if you take a biological tissue and you perform your cryosectioning onto your moldy surface, then you can have actual external calibrants that can be used to generate a calibration curve. Another model would be to spot the calibration standards across the tissue themselves and use that on a control section and then be able to compare your sample section. And then Reed very nicely overviewed the tissue mimetic model, which allows you to also perform quantitative analysis. So while I'm not here to certainly tell you which method is the most appropriate for your own application, it's important to kind of consider the different aspects of these tools if you're wanting to do a true quantitative analysis. This has also been summarized quite nicely by doing in this reference paper. So again, focusing mostly on MALDI, but it's important to address that other strategies for mass spectrometry imaging are available. And including, you know, things like dilution series on top of the control tissue, which I just showed, or underneath the tissue, as well as, you know, spraying or applying the internal standard across the tissue section itself. So at GSK, they did a very nice multi-center study where they looked at the distribution of clozapine that was applied by perfusion and then coated the tissue with D8 clozapine and wanted to determine the concentration of both clozapine and its metabolite. So this very nice study then also had 3 different analysts that were looking at the tissues and was able to report kind of how reproducible and similar these studies were. And so just to walk you through this study, and certainly I think it's, like I said, great to have Reed here, because if we open this up in the Q&A, he was part of the study, he can certainly answer more questions than I likely can. But what I'd like to just kind of point out is that the blue and is the mimetic model without normalization, the orange is with normalization, and then the purple is the dilution series with raw data, then followed by the normalization. And so in all cases, right, we see that the normalization can vastly help improve both the variance and the accuracy of the measurement. And so for relative comparison, the LCMS homogenate is here shown in the green on the right. And so also, you know, you see there is, you know, some differences from analyst to analyst. This is the clozapine on the top, this is the metabolite on the bottom. But nonetheless, what this shows is really, I think, the importance of considering internal standards, especially when you're trying to perform, you know, absolute quantification by imaging. Another aspect just to look at, this is the section, the image that shows the raw data and then the normalized to the D8 internal standard. And then here's internal standard that's shown as well as the normalized on the right. So again, if you're wanting to do, you know, kind of absolute quantification by mass spectrometry imaging, you know, the appropriate internal standard is highly recommended. So in conclusion from, you know, this study as well as others, you know, the accuracy was reported at about 80% compared to LCMS, and I think really takes home this need for standardization. Many different groups have attempted to look at reproducibility and standardization for mass spectrometry imaging, and, you know, including an internal standard will greatly assist in that. And that these imaging protocols can certainly provide results that are similar to LCMS. I think what's really an important takeaway message from this study and others is really to think about what the need is for you know, absolute quantification versus relative quantification, and how that might play a role in the workflow that you use for your own independent analyses. So with that, I'll just end on a quick little plug for those that might be in the Midwest to save the date for July 7th for our Chicago Mass Spec Day. And I'll conclude there, and I believe we're going to open it up for some discussion. Thank you.
Great job, Stephanie. Thank you so much for that. So if anyone in the audience has questions, please put those forward now in the Q&A. So there's a couple questions that I've created just for our future audience. One of my questions is for Reed, and Stephanie can chime in, is how long does it take you, Reed, to develop a new imaging method for a new drug. I've heard numbers and I was like, oh my, I don't think I can do that in my laboratory. Do you want to
Well,
comment on how
sure.
long it takes?
Yeah, it's a good question, and it does depend. Normally, we can look at kind of the structure of the drug and get an idea of maybe some of the basic parameters, positive mode, negative mode. But unfortunately, it's also sometimes it's just kind of, I guess, empirical. It'd be great if we could go much further than we actually do in terms of looking at a structure and saying, well, this method would be ideal or not. So it's very much, we have a starting point that we typically would use for positive mode, or negative mode would be a different one, but a similar starting point. And then from there, we use the mimetic model quite extensively to do sample optimization, because it does provide you with a nice source of a way to kind of know different concentrations in a relatively homogeneous distribution. So it is a pretty effective sample to look into different methods. So I'm kind of rambling. The short answer is it's something would like to have more systematic. Right now, we have various matrices we can try, but it's very much usually just creating the mimetic model and then adapting the approach, different solvents, matrices if needed, to try to maximize that sensitivity. So.
Okay. Any favorite matrices that you start with for both positive and negative ion mode for drugs?
For drugs, we primarily use DHB for positive mode. I'd say we very rarely shift from that, maybe in some unique circumstances. And then with negative mode, it's more of a challenge. I mean, it's we've successfully used norharmane, we've used 9AA. I think most recently we've had pretty good success with norharmane. So, but I would say generally speaking, it's the negative mode's more of a challenge, where sometimes we can struggle. So, but those two have served us well in a couple studies.
And then what about you, Stephanie? Do you have a strategy for one of my questions was actually, when you're looking at the lipids for quantification, do you have a specific matrix that you go after for your lipid imaging? And then second, do you find that there's different strategies that you need for different classes of lipids?
Yeah, I think in our space, it's a little bit different because there has been so much done in MALDI for the analysis of lipids. So, we can build a lot from the literature there. It's a little less empirical than I think Reed has to deal with drugs. But we use a lot of the same ones, right? 9-aminoacridine, DAN, and so forth. And there have been quite a number of extensive studies that show, kind of preferential ionization of certain lipid classes with different matrices. So we certainly build on the literature for that. But I wouldn't say that there's necessarily kind of a workflow that we use. Now, if we do find something that, let's say we go the opposite direction, today I showed where we start with imaging and then we go to LCMS, but if we were to maybe find something by LCMS, so we wanted to actually map spatially, then in that case, we would kind of do a much deeper dive to look at the different matrices and the performance for that analyte in particular.
Do you both apply matrix what's your methods for applying matrix? Do you do spraying or do you do sublimation? Stephanie?
We do both in my lab.
Okay. And then Reed?
Primarily spraying, but
Okay.
in some instances for high spatial resolution, we would use sublimation as well, yeah.
Okay. Do you do derivatization for drug detection, Reed? So how frequently does that have to happen?
Not frequently, but that's almost kind of a last resort if all else fails. So we have done that. It's been quite a while, but we did do that successfully, and it did seem to work. But it's obviously adds a pretty extensive element of additional effort and sample prep consideration. So I think there's potential there. I mean, I think obviously others have shown that as well. So I guess it's kind of something we have in our back pocket, if you will, but we certainly don't use it very often at all. It's very rare, I would say, but kind of a last resort.
All right, thank you.
Yeah, I'll ask one for Reed. So Reed, you spent a lot of time showing the multi-omics too, the lipid, protein, and endogenous metabolite profiles. And those were great for looking at structure markers in terms of what you were looking at tissue. Have you also been able to look at that and figure out new mechanisms of toxicity or drug metabolism?
Yeah, that was kind of one of the final slides that I skipped, but I think that's the big push. So we've primarily been focused on drug distribution, but the next kind of frontier is now opening that up and really, I guess, more systematically incorporating these endogenous changes, because there's a huge opportunity there, right, to look at the mechanistic changes that drive efficacy, toxicity, and I think it's a huge opportunity. And once again, we're really putting a lot of effort into starting to build out those analysis methods and the data processing behind that. But no, that's kind of where we are. We're really pushing down that approach now to really better incorporate some of those endogenous change measurements into also looking at the drug distribution, but in correlation with endogenous changes. So, yeah.
That's great. Yeah, getting that spatial information with it and those small little regions on how it's metabolized is going to be really cool.
Yeah, definitely.
Okay. Are there any questions? Reed, do you have questions for Stephanie, or Stephanie vice versa? We're right at the limit of our time.
Well, no, I definitely enjoyed Stephanie's talk, and I think she did a great job giving an overview of all the different considerations. So, but I won't drag us out any further than I already have.
Yes.
My thing.
Well, thank you both for excellent talks. Thank you, Kristen and Conference Solutions. And so that concludes our Spatial Omics session for this month. So thank you.
Thank you, everyone.
Thank you.
Bye.